Welcome back everyone to the cell signaling course. We had discussed earlier that what are different types of signals, ligands, stimulus, agonist, whatever you tell, because that is one of the first and foremost component of the signaling. We have also understood that we need at least a receptor which binds to the signal and thus activates the process of signaling. Now in today we will see that how different types of receptors are available in the living world and how do they function, how do they bind to the signal, and what next happens to them. So as we have understood that receptor is present on the receiver cell or a sink where ligand should bind and culminate in the process of signaling. So a signaling receptor more often than not is a proteinaceous multi-domain protein. It has multi-domain, which means that they are arranged in a fashion so that the activity which is desired of the receptor is met. So a receptor which is multi-domain has an organization which should let it perform at least two main functions. What is very obvious that the signal which is available in surrounding, receptor must bind to it, right? So it should sense the signal which is coming from the extracellular environment, whether it is directly binding at the cell surface or the signal permeates inside the cell and binds the receptor over there. Now this extracellular signal which has been sensed by the receptor must activate something so that the receiver cell knows the presence of the signal. In that case, it should communicate accordingly the changes in the intracellular environment. What I meant here is that if this is your membrane, this is your receptor. So it should be able to bind to the signal. Consider this as your signal. And once it binds to the signal, it has sensed the changes or the message which is in the extracellular environment and then somehow transmit this inside the cell in the intracellular environment. Now when we further look into the receptor, very much like what we saw for the signals, the receptor can also be of different type, different functionality, or different properties. Like here, as we saw, that signal could be soluble or it could be membrane bound. We have also understood that receptors can also be soluble or membrane bound. Again, I would reiterate that soluble receptors are present inside the cytosol and they bind to the ligands or signals which come inside the membrane, crossing that barrier. Whereas membrane bound receptors, they prefer binding to the water soluble molecule which cannot permeate inside. So the receptor must bind to the signal. That's the very first thing it has to do. Otherwise, it's not a receptor. That is why often you have a cognate pair of signal and the receptor. So first and foremost aspect of the signaling through receptor is the binding of signal either directly or indirectly. Sometimes you need a mediator molecule through which the signal binds to receptor, or sometimes signal comes with a property which helps bring receptors together and thus facilitate the binding. Second aspect is that once it has bound to the receptor has bound to the signal, it must activate the process of signaling. Here, more often than not, activation is associated with the receptor itself, where it either opens up so that extracellular environment message is conveyed to the intracellular system, or it undergoes a conformational change. When we say conformational change, proteins are arranged in their three-dimensional manner where domains are interacting or put forth to each other in such a way that they are inactive. Once the signal binds, it must undergo certain conformational change which activates it, which lets the signal go inside. If you remember from previous classes, in such cases, when receptor is membrane bound, signal does not enter inside the cell. So in this case, signal binding to the receptor must activate something inside, and that is what is the third thing which the receptor should achieve. Basically, the binding of signal in the extracellular domain of the receptor, there must be a message which is sent inside. One other thing which is commonly seen with receptors is that they undergo a process of dimerization, and this is where some receptors are dimeric by default and some receptors undergo dimerization when facilitated by the presence of a dimeric signal molecule. This dimerization should initiate a cascade inside the cell, downstream activation, which will allow passage of molecule in case if there is a receptor which is a channel, or it should allow the movement of certain proteins which are effector proteins. In the previous class we saw there are signaling molecules, there are effector molecules. So these molecules must be activated, and the passage of molecule should be allowed only when the receptor is of channel type, which can allow entry of small molecules and ions and so on. So basically in this case what will happen is that your receptor, which is again here seen as a membrane bound thing, if I can say this is a dimeric molecule, it binds to the ligand and then activates the system, which means that signaling proteins, and then this signaling protein will activate the effector protein. And what we also know that sometimes these molecules can be classified as messengers, and these messengers will carry the message which is present in the extracellular environment to the intracellular environment. And in the previous class we saw that how it could mean that cell moves by cytoskeletal rearrangement, or it can activate the transcription process, or it can activate certain enzymatic activity. Now this membrane bound receptors which have multiple domains, they span the membrane multiple times or just once. It's the property of the receptor. So the membrane spanning is something which is very important to hold the receptor stable in the membrane. If you do not have the membrane spanning domain, basically it will not serve as a receptor for those molecules which cannot gain entry inside the cell. So the receptor, as I said, they can pass the membrane once, more than once, and some specific number of turns. So like here if you see, this is a channel type of receptor. It can pass multiple times this membrane. Or you can have a receptor where you can have the receptor as a different entities as heterodimeric or homodimeric molecule. Some of these receptors can have two distinct types, and they need to come together to activate the signaling process. Here you can notice that the signaling happens when receptor comes together, and one more important thing to note is that the receptor is passing the membrane. Receptor is passing the membrane only once. But in unique situation of a set of receptors which are called GPCRs, the receptor passes the membrane 7 times, which is basically helps them be characterized as 7-pass transmembrane receptors. This also gives them a serpentine appearance, or like a snake. That's why they are also called serpentine receptors. So every receptor which is present on the membrane waiting for signal to bind has unique feature, but what we understood is that there is at least an extracellular domain where it can bind to the signal, and an intracellular domain where it can bind to effector molecules or signaling molecules, and definitely a membrane spanning domain. Now some of these receptors, I spoke earlier that some of these receptors could be channel type, so they just allow the passage of the molecule through the channel. But some of these receptors, they can have enzymatic activity. What enzyme activity which we say? And more often and as needed, these activity is actually located in the C-terminal cytoplasmic tail. Why? Because ligand binding is happening in the extracellular domain, membrane spanning domain is holding the receptor in the membrane. You need this message to be conveyed inside the cell. That is why cytoplasmic tail is where activity is ingrained. So what are these activities which can be there? Either this receptor can further be associated with an enzyme which can generate the signaling, or it can have an integral activity as mentioned here. It can actually generate a cascade only when it can activate the process, whether by directly having the intrinsic enzymatic activity or binding with the protein which carries or brings the enzymatic activity. The most common enzymatic activity as known in case of many of the receptor is to facilitate a phosphorylation dephosphorylation cycle. You understand that phosphorylation is attachment of a phosphate group, and dephosphorylation is removal of that. There are specific enzymes which catalyze these processes. More often than not, you see many of these receptors have kinase activity which helps in the attachment of phosphate group. But you need a dynamic process of attachment of phosphate and removal of phosphate. That's why there are activities which are associated as a phosphatase activity with these receptors. Also, sometimes these receptors, they may not have intrinsic activity, but they do associate with molecules which help in the process of GTP binding and hydrolysis. So GTP can be hydrolyzed into the GDP, and the energy which is released, that is used for change in the receptor conformation or recruiting new molecule which will carry the signaling further ahead. Now if we look at the functioning of the ion channel receptor, the ion channels, as I mentioned earlier, they could be multipass transmembrane receptor. They have an opening for ions to pass, but if you talk about channels which are not leaky type, meaning they do not randomly allow ions or small molecules to move through, then they generally are present in a closed conformation, the closed channel, which does not allow any movement of ion. The movement of ion is basically sometimes for receptors bidirectional, or it could be unidirectional. So here consider the situation where the signal is present in the extracellular environment. You have the plasma membrane and the ions are ready to move inside. However, the signal is not available, or the trigger for activating this movement is missing. When that signal is available, you will see binding of this ligand happens to the receptor, which brings the change in conformation of the receptor, which now makes this channel more open type. Now once the channel is open, the desired small molecules like ions, they move inside, and this brings the change which is required in the intracellular milieu and allows the signaling to move forward. The other type of receptor which I showed you earlier was this GPCR, G protein coupled receptor. What it means that this receptor is associated with another protein which is called G protein. So here is the receptor, and if you remember, I told this is like a serpentine receptor because of its shape, and it also passes the membrane 7 times. So that is why it is 7-pass transmembrane receptor. And the G protein which it associates with is present in the vicinity. Normally, when signaling is not happening or in the absence of proper ligand or signal, the receptor is separated from this G protein. When the signaling starts, you will have ligand binding, like here, this is your ligand which has bound to the receptor. This will bring a change in the receptor conformation itself. And this conformation will allow the association of this G protein with the receptor. So this class of G protein are called heterotrimeric because you can see that there are 3 units over there, G alpha, beta and gamma. When the signaling starts, ligand binding happens here in the extracellular region of the receptor. Internally in the cytoplasmic tail, here you will see G alpha moving and binding to GTP. This dissociates this trimeric complex. G alpha goes and binds to the GPCR, and alpha beta gamma complex remain here. Now both of these molecule, whether it is G alpha associated with the receptor and beta gamma which is remaining in the membrane, are free to do the signaling process. So what you can understand is that here this 7-pass transmembrane receptor will bind to the ligand and it will activate the system, and the first part of activation is recruitment of G alpha. Now this G alpha, as I said, these are another type of enzymatic activity which is associated with the receptor. It will bind to GTP and hydrolyze it to GDP. This energy which brings the conformational change as well as recruits next set of molecule, you can say signaling or effector proteins. Also here sometimes the next molecule generated is called messenger, secondary messenger. So what you notice is that in this case receptor when binds to ligand, it allows the binding of G alpha subunit to itself. This G alpha with the activity of GTP hydrolysis allows activation of next set of molecules, whether it is signaling molecule, second messenger, and this is what will allow the signaling to move forward in the desired direction. Then you have the enzyme coupled receptors. What are these enzyme coupled receptors? So here you can see that receptor is present, and these are mostly of single pass transmembrane type receptor. Here these receptors could be either homodimer or heterodimers. Looking at the structure, you can suggest that in this scenario this is a heterodimeric situation. This is, one can say, 1 type receptor, and this you can say 2 type receptor. Also you can say that it could be homodimeric molecule eventually, right? So what you notice here is this thing which is the kinase domain. Kinase is an enzyme which will utilize ATP, hydrolyze it into the ADP and phosphate. This phosphate which is generated will be transferred to a particular amino acid or target residue. We know that this phosphorylation event is very important, and this is what will trigger the signaling to move forward. So the single pass transmembrane receptor which we see here, when it binds to the receptor, it will probably get into something as a very preliminary step of signaling which is called dimerization. So in this class of receptors, whether it is single pass transmembrane receptor which are responding to the growth factor or other molecules which we will see in later classes as we go forward, that they need a dimerization event so that signaling can move forward. So what will eventually happen in these cases? The ligand will bring the receptor there. So in this cases, ligand when binds, it brings the 2 type of receptor together. Remember, the 2 types could be same or different. When they are same, it is homodimeric situation. When they are different, it is heterodimeric situation. So what generally will happen when the ligand binds? What it ends up doing is reconstitutes the dimer. and this is one of the first step towards the activation of the signaling. So this activated receptor, which has this enzyme domain over there, I told you that this is a kinase domain. What it will do, it will phosphorylate the target. It will attach the phosphate group. I told you that ATP will be broken down into ADP and the phosphate will be added there. So this phosphate, when added, makes the molecule either active or inactive. And one of the first thing which is seen often in most of these receptors is that they phosphorylate themselves at multiple residues. Don't be confused here that it is phosphorylating in a residue which is in the membrane. I am trying to show that multiple location the receptor itself can be phosphorylated, and that activity continues as long as the receptor is active. So it will keep utilizing ATP and converting into the phosphate group. So this molecule which is finally activated, this will have targets. Often these molecules are kinase themselves, so they will perpetuate this signaling event by activating next set of molecules. Sometimes these are, like we discussed earlier, that these could be a cytoskeletal protein which will get modified and then it will activate the system so that cells can migrate or cell can move from one place to another place or make some projections which are coming out of the cell surface. Or it can be the enzymatic activity of a metabolic enzyme which is regulated. Now, like I said, often these activity is associated with the receptor. What also is seen sometimes is the phosphatase activity. You would appreciate that here in this process where receptor is getting phosphorylated and activating things, if it continues to be in active state, it will be a problem. So you need to inactivate. So there will be a set of molecules which carry the phosphatase activity to regulate the signaling, a feedback mechanism which will regulate the signaling mediated by such enzyme coupled receptor. In addition to that, what I meant from this particular statement is that receptor itself can have phosphatase activity. Rather than kinase activity, they can have phosphatase activity. And once this molecule which is phosphorylated, it can have thousands of targets. And we will see in coming classes that what all places where phosphorylation can actually happen and how that leads to the further activation of the signaling. So if I summarize today's class, what we have understood is that receptors, similar to the ligands, they could be soluble or membrane bound type. Based on that, they will initiate a type of signaling event inside the cell. They can carry the integral enzymatic activity of their own, or they can associate with molecules which have enzymatic activity. And we saw often the kinase is the prominent enzymatic activity which is associated. We also saw that there could be a GTP binding and hydrolysis kind of activity which is associated with receptor. The membrane bound receptor, they span the membrane either once or more than once. We saw cases where they are spanning as many as 7 times. So what you would have understood by now that receptors could be classified based on how they appear, what kind of domain they carry outside for signal binding, what kind of domain carry inside for enzymatic activity, and how many times they are passing the membrane. And one of the most important thing which I mentioned over there, the binding of signal to the receptor must activate the receptor, and one of the earliest step towards that is the dimerization of the receptor. So in a normal signaling event, ligand should bring the 2 copies of the receptor together, dimerize it so that receptor can be activated. And this is what will initiate the cascade, which will take the signaling in the direction depending on what was the ligand, what is the cell type, and what is expected out of this process. Thank you.